Let me go ahead and
Excellent.
share the screen as well.
Sure, great. Excellent. So welcome everybody to the Cancer Genomics Cloud webinar series for February. I'm very excited today to welcome Dr. Varduhi Petrosyan to our webinar series. She's from Baylor College of Medicine, and she's going to be talking about recent work she and her colleagues have published using the Cancer Genomics Cloud to run a combined network-based analysis for metabolomics called Connect the Dots with WGCNA, or weighted gene co-expression network analysis, to find biomarkers for disease. So without further ado, take it away, Varduhi.
Okay, I'm going to start with the presentation. So the title of this talk is the same as the title of our paper, which is Identifying Biomarkers of Differential Chemotherapy Response in TNBCs, which are triple-negative breast cancers, using patient-derived xenograft and a combined CTD-WGCNA approach. So this is the graphical abstract of our paper, which breaks down our analysis. So we start by first modeling triple-negative breast cancers in immunocompromised murine mouse models. We then generate two cohorts, one that's going to be a control cohort and injected with vehicles, from which we derive baseline gene expression. We also treat these murine models with a variety of different chemotherapy treatments and use this treatment response in addition to the baseline gene expression to build these weighted gene co-expression networks. What we then do, and really the addition of this paper, is we use CTD in combination with the WGCNA approach to successfully identify small and informative multi-gene biomarkers of both platinum and taxane chemotherapies. We then show that these multi-gene biomarkers are informative for both other PDX cohorts as well as human clinical trials in determining the response of the patient or the PDX to these drugs. So we study breast cancer as breast cancer is still a current leading cause of cancer mortality. It remains the most common cancer in women, with 2.3 million women a year being diagnosed with breast cancer. So there has been a huge improvement in the treatment options for several of the subtypes, but some subtypes, such as triple-negative breast cancer, still don't have a good targeted therapy available. So breast cancer is a highly heterogeneous disease, or set of diseases, if you will, and going from the one with the best prognosis to the one with the worst prognosis, we have the luminal subtypes of breast cancer, which are going to be the most common and have a lower level of cancer growth. So these are going to be positive for the hormone receptors, estrogen and sometimes progesterone receptors, and negative for the HER2 amplification typically. So these luminal cancers have a targeted therapy, which is tamoxifen, that I'll be discussing in depth a little bit later. We also have normal-like breast cancers, which are less common, but also have worse prognosis than, for example, luminal A, and our HER2-enriched breast cancers. So these are a subtype of breast cancers that's going to be negative for the hormone receptors, estrogen and progesterone receptors, but it's going to have a HER2 amplification. So this HER2 amplification increases the growth of these cancers, and they're going to be having a worse prognosis than the luminal-like cancers. These cancers also have a targeted therapy that's widely used, which is Herceptin or trastuzumab. Today we'll be discussing mostly triple-negative breast cancers. So these cancers make up about 15 to 20% of the breast cancers that are diagnosed in the clinic, and they're found primarily in younger women and mostly in African American women. There is currently no targeted treatment, and as you can see here, triple-negative breast cancer has a poor prognosis to this date. So while the survival times of the other subtypes are increasing, we still have this gap here in the survival of patients with triple-negative breast cancer. So one of the reasons triple-negative breast cancer has a poorer prognosis compared to the other subtypes is that if you come into the clinic with a luminal breast cancer, you're likely to be treated with tamoxifen. So tamoxifen outcompetes estrogen for this estrogen receptor and slows tumor growth. Likewise, if a patient presents to the clinic with a HER2 subtype, Herceptin is likely to be the first course of treatment. So Herceptin is going to outcompete and bind to the HER2 receptors and again slow cancer growth. Unfortunately, while there are several different drugs in development for triple-negative breast cancer, we don't have one that's commonly used for primary triple-negative breast cancer. So we have, for example, the PD-1, PD-L1 inhibitors. So this is going to inhibit the signal between PD-1 and PD-L1 and allow T cells to better target the tumor. We also have in development PARP inhibitors that are, such as Orofin, that are going to be targeting this DNA repair mechanism, which leads to the breakdown of DNA. And there are other inhibitors that are currently in clinical trials, such as antibody drug conjugates. So some triple-negative breast cancers are treated with these therapies, but currently the FDA has said that to be treated with these therapies as a first-line approach, you either need to have metastatic breast cancer or a very advanced and very aggressive local breast cancer, triple-negative breast cancer. So unfortunately, for now, the standard of care for these triple-negative breast cancer patients remains a combination of chemotherapies. So this can be up to 8 to 10 different chemotherapies working together. The problem we have is that only 25 to maybe 30% of triple-negative breast cancer patients will achieve pathologically complete response to each single therapy. And because we're layering different chemotherapies on each other, we're increasing the morbidity and we're increasing unwanted side effects. So what we'd like to do here is to find a way to identify for each specific chemotherapy which patients are going to be responsive versus resistant for each therapy. And the way we do this is through the use of PDX models. So PDX models are patient-derived xenograft models. And these are formed by taking breast cancer tissue from a patient and implanting it into the mouse. These are then generated into larger cohorts, which we can treat with either vehicles to see, for example, baseline gene expression, or different drugs to compare the treatment of different therapies. We can also use these PDXs to identify particularly resistant tumors and to see if there's any genetic mutations or gene expression changes that we can identify that are present in the resistant mice and not in the responsive mice. So this work is part of a larger project in our lab where we incorporate patient-derived xenografts, patient tumor data, deconvolution, as well as a network approach to really investigate breast cancer biology and look specifically for biomarkers of response. What we're going to be addressing today is using these patient-derived xenograft mirroring mouse models, as well as network analysis to identify a small but informative subset of genes that you can use to predict response to 2 different therapies, taxanes and platinum therapies. And here, for those of you that are joining us, I'd really like to point you towards the BCM PDX portal. So this has a wealth of PDX data and it's commonly available. So the numbers here are for all of the publicly available PDXs. If you're interested in PDXs that aren't publicly available, you can email our collaborator, Mike Lewis, who runs the PDX core, who can get you information on those that aren't publicly available. So the interesting thing here is that for each PDX, we have information, first of all, on the patient, their vital status, exactly what kind of breast cancer they came into the clinic with, what the course of treatment was, when each treatment was given, and when each PDX was made. So for the PDX model, you're also going to have all of the hormone receptor statuses, the BRCA status. You're going to have RNA-seq data available as well, and the treatment for each of the models and which specific strain these PDXs are derived from. Likewise, for most patients, you have, and in PDX models, you have concurrent histology information, so you can check that, in fact, the hormone receptor status is going to carry over from the patient to the PDX. And our talk today is going to be primarily focusing on looking at biomarkers of chemotherapy response, but quite a few of these patients in PDXs also have metastasis information. So if you're interested in looking for markers of, say, metastasis in triple-negative breast cancer, this is also a good resource. So we start first by asking how well does our PDX collection recapitulate the breast cancers in, say, TCGA, the primary human tumors. And this is just a qualitative look at the copy number alterations between our triple-negative PDX models and our TCGA models. And just kind of quantitatively, you can see that they have similar patterns of copy number alteration. We also looked at mutations both in our models, PDXs, as well as TCGA. And what we see in this figure here is that there are similar mutational profiles between our PDX murine models and the TCGA breast cancer tumor. On the right figure here is going to be specifically which kind of mutation types occur in our PDXs versus the patient. This is looking specifically at our triple negative breast cancer PDXs, and we see that the profiles here again mirror those of the patient. So for these PDXs, we have deep RNA-seq at a level of 220 million reads per sample. And what we use this for is also to see how well the transcriptomics map between our PDXs and our primary tumors. So a question that's still in the field is, what exactly are you modeling with your PDX models, fly models, what have you? And what we can see here is that our PDX models are primarily modeling a basal, which is in red here, group of TCGA tumors. And we do have some models here that are modeling a more luminal-like subtype. So similarly, we have proteomics data from about 8,000 proteins, and using the 1,000 most variable plurities to cluster this data, we're able to see again the PDXs are interspersed primarily among basal human-like tumors. Basal-like human tumors, my apologies. So what we also have is, as well as this deep multi-omic profiling, we also have the response for all of these PDX tumors, and we have quantitative responses as the log-fold change. So over the course of 2 weeks, how much does the tumor grow or shrink given therapy? And from these log-fold change values, we then classify the tumor as pathologic disease, stable disease, partial response, or complete response. And what we can do here is, because we also have the data for the patients and what they were treated with, we can then ask, how concordant is the PDX's response to therapy with that of the initial patient? So here we're looking at the docetaxel treatment of PDXs. So D20 stands for docetaxel treatment, 20 milligrams per kilogram treatment for these mice for 2 weeks. And what we're able to see is that even when the patient treatment doesn't quite correspond to what we used on the mice, so these are all taxanes that the patients are treated with. But for example, as you can see, some of the patients are going to be treated in combination with docetaxel plus doxorubin, or they're going to be treated with paclitaxel. So although the treatments don't correspond one-to-one between the PDXs and the mice, we have an 85% concordance between how our patients are responding in the clinic and how the mice are responding to treatment. So because of this high concordance between our PDX models and the primary patient tumors, we then hypothesized that we could use this PDX cohort to identify biomarkers of response to therapy that could be carried over to the clinical setting. So how do we start interpreting these complex data sets? We have quite a large number of RNA-seq expression profiles at a very high depth. And what we can do here is we can build networks to identify patterns within this transcriptomics expression, and then to also use the chemotherapy treatment information we have to identify patterns that are going to be specific to resistant versus responsive mice. So the standard approach to this network analysis is a program called WGCNA, or Weighted Gene Co-expression Network Analysis. So this has been widely used across the field in diverse applications, anywhere from fungal research to looking at, for example, Alzheimer's disease. The problem with this approach is that WGCNA clustering uses hierarchical clustering, so it builds this tree system of how the genes are related, but it cuts at a static level. So you can pick as a user where you want to cut your tree. The problem here is that the modules that WGCNA creates are very large, so 30 to 200 genes in size in our particular data. And thus, for WGCNA to be able to correlate these modules to your phenotype of interest, it uses something called module eigengene. So it takes the signal of each module and it flattens it into 2 principal components. And it then correlates these principal components to your phenotype of interest. The problem with this flattening and correlation analysis is that you might miss genes that are very informative, but they're not going to contribute significantly to the WGCNA module eigengene signal. So the modules that WGCNA produces are too large to be easily biologically tractable, especially when you're working with smaller sample sizes. So for example, we're going to have 45 PDXs in the data we're talking about. If you have 200 genes, you're going to be underpowered to identify informative ones easily. And using the module eigengenes flattens the signal, and it makes it difficult to determine which specific genes are going to be giving the signal in an informative module. So here we are applying a technique developed in our lab called CTD to connect the dots. and identify highly connected nodes within a weighted graph. So going from left to right, if you're looking at the purple nodes which you're interested in, you're going to have highly connected nodes which have a smaller p-value. And then the figures on the right, we're going to have nodes that are not as well connected and have a larger p-value. So what CTD does is it uses the algorithmic significance theorem and data connectedness models to see how you can present these modules as being highly connected. So the modules are scored if they diffuse from each node. So this is going to be a hit, and a hit. If you're trying to encode this module, you're going to have a longer module because you're going to do a hit, a miss, and a hit, and so on. So the more concisely these modules are encoded, the smaller p-value is applied to each module. And what we can do now is we can input a weighted graph. So for example, if you have an experimental and a control graph, as I was mentioning, mice that are resistant and responsive to particular chemotherapy, we can build a weighted graph that's going to show the differences in the gene expression between these two. And then you can input your nodes of interest, which in our case are the large WGCNA sets. And what CTD allows you to do is to identify these highly connected specific sets of nodes. So our hypothesis going forward in this paper is that these highly connected sets of nodes may be biomarkers of response, as we build two graphs for each model. We have one that's resistant and responsive PDXs, and one that's just built on the responsive PDXs. So we're going to prune our general graph based on the one generated from the responsive PDXs. And what we hope to have left is signal that's specifically related to chemotherapy response. So by identifying these highly connected subsets, we are then able to identify gene markers of resistance versus response, as we'll be showing later on in the paper. So for our two conditions, we have treatment of these mice with both carboplatin and docetaxel therapy. We're able to identify small multi-gene biomarker sets that are going to be informative for treatment response. For carboplatin, we identify 9 informative genes, and for docetaxel, we're able to identify 6. So another point I'd like to make here is that if you'll see the cell types on this column to the right, for both carboplatin and docetaxel, as well as identifying informative epithelial genes, we also identify informative stromal genes. So what this allows us to see is that even in this immunocompromised murine model, we're able to see their stromal signatures of resistance and response, which are then going to be mirrored in the human patients. So one of the questions we asked in the study was, how do different feature selection methods work with one another? So given the same data that you input, using a different feature selection, will it give you similar genes or a highly diverse set of genes? And what we find here is that each feature selection method will give you a very distinct set of genes. So if anyone needs help reading these graphs, I know they're a little complex. What we have here are the distinct intersection sizes. So for example, if we're looking at our CTD method, which has 9 genes for carboplatin, we're able to see that 5 of these genes are distinct. 2 of these genes are going to be common between our CTD method and WGCNA hubs. So these are the central genes in these large WGCNA modules. And 1 gene is going to be common between the CTD, WGCNA hubs, and mutual information test, an F test, a Bruda test, and the recursive feature extraction. So you're able to see how much of these genes overlap with one another and how many of these genes are distinct between each method. So what we then would like to do is ask, how does the CTD method compare to other commonly used feature selection methods? So using a root mean squared error test, what we see is that the CTD method in both carboplatin and docetaxel will give us the smallest root mean squared error. So it's the most accurate in predicting resistance versus response in our specific PDX cohorts. And what we then wanted to see was that, so our method is a combination of a previously used WGCNA method. And what we wanted to see is, are we adding information on top of what can be attained just using WGCNA? So here we're comparing our CTD WGCNA method in red to the WGCNA hub genes. So once you extract informative modules, these are the genes in the center of these modules, and the large WGCNA modules. So in both, this is showing carboplatin, for example, and CR stands for complete resistance. CRPR means that the mice are complete or partially complete response, my apologies. And CRPR stands for complete and partial response. So what we're able to see is that by adding CTD to this previously used WGCNA method, we're actually able to add information and get better predictions with smaller amounts of genes in both cases. So then we asked if we could predict for each specific PDX model what the best therapy for each of these mice was going to be. And here we're looking at carboplatin versus docetaxel therapy. So for each PDX, we're predicting how sensitive the mouse is going to be to carboplatin, how sensitive the mouse is going to be to docetaxel, and then making a call one way or the other what the best therapy for this PDX would be. So here, the dots in red indicate that docetaxel is the best therapy, and we've correctly identified it. The dots in blue identify that carboplatin is in fact the best therapy that we've correctly identified. And the black dots means that we've correctly selected the therapy. So overall, we have an accuracy of about 73% in identifying the best therapy. But because these aren't qualitative reads, this is actually going to give you a predicted log-fold change. What you can also do is you can ask, are these log-fold changes very different to each other, or are they very close to each other? So if we add another layer and say that we predict that these mice will be very different in their response with carboplatin versus docetaxel, our accuracy for predicting the best response actually goes up to 85%. We then asked if these multi-gene biomarkers that we predicted are going to be informative for other chemotherapies in the same class. So these multi-gene biomarkers are trained on either docetaxel or carboplatin resistance versus response. And what we're looking at here is another data set. So this is going to be a data set from the Rosamond Goodman Cancer Research Center. And what they have here is they have response of PDXs as complete and partial response or complete response for both cisplatin and paclitaxel. So using our biomarkers, what we're able to see is that we have some signal when it comes to predicting both the paclitaxel and the cisplatin responses, particularly if you're looking at complete response versus all else. In this other PDX dataset using different chemotherapy agents, which are under the same umbrella. So we then asked how predictive these biomarkers are going to be across platforms. We trained these biomarkers using deep RNA-seq data, but some of the older studies use, for example, array data. So using a PDX array dataset from the Curie Institute, we're able to see that we also have signal when predicting across just, in this case, 9 genes, the resistance of response using array data. So most importantly here, I think, we're also able to see that these small sets, so 9 for cisplatin and 6 for paclitaxel, or we trained it on docetaxel, are predictive in patient response to therapy. So this is, on the left, is data from the Dana-Farber Cancer Institute, and they have here a patient array that has and also response to cisplatin. So even in the array data set and even in patient instead of PDX cohorts, we're able to see that we have signal for predicting the response or resistance of these patients to cisplatin. Likewise, we also tried this method in a brightness patient data set. So here they have patients that were treated with paclitaxel followed by AC treatment. So already we're going to be kind of complicating the results because the responses aren't after paclitaxel, but after concurrent treatment. But even with the combination of these two treatments, we're able to see we still have some signal using just these 6 genes in this patient data set. So as a general conclusion, PDX models, we've shown recapitulate primary tumors. We've shown that they have similar mutational profiles, transcriptomic profiles, and proteomic profiles. And then we've shown that we can use CTD to connect the dots, as it were, in these large, very complex graphs and identify these small biologically informative sets of genes, which in our case can be used to look at resistance and response of tumors. And we found that biomarkers of response, both for our platinum agents, which were predicted on carboplatin, and for our taxane agents, which we use docetaxel to predict, are going to be informative across agents of the same class, across platforms. So you can use these biomarkers on both RNA-seq datasets as well as array datasets, and are going to be predictive in both PDX models in other cohorts, as well as primary human tumors. So before I go into more how we use CTD and how you can use it on the cluster, does anyone have any questions here?
I don't see any questions in the chat or anything. I do have sort of just one small question. How did you define your hub genes for WGCNA? It's one of the things I always kind of struggled with.
Sure. So once you, if you'll allow me to go back just a few slides. So this is kind of the initial output of WGCNA, right? If this looks familiar to you, they have the modules, how correlated they are. So once you identify the modules you're interested in, so say we're going to be interested in this trichoid module for percent change in baseline response. So this is just a data set that has to do with, I believe, alcohol intake. But once you say that I'm interested in this trichoid module, there's then a centrality function with WGCNA. So these hub genes are going to be the ones that are going to be most central to your module. And you can apply another filter and say they're central and are they related to this phenotype as well, right? So WGCNA does allow you to extract the central genes, but you're not going to get all of them formative ones potentially.
Okay, cool. Thank you.
Sure. But this is also important because if you'll see here, if you're trying to just look at the WGCNA modules, they're going to be very large. So for carboplatin, you're going to have 97 genes, and the hub genes allows you to get down to 14, right? So you're trying to kind of extract the signal there. So any other questions, or are we okay to proceed perhaps?
Yeah, I think we're okay to proceed.
Sure. So CTD is actually publicly available on GitHub at the moment, and you can install it with either R or Python. If you're going to be doing a local install, I highly recommend using the Python version as it's more up to date. It's also available as a Docker app, but I think excitingly, we can also use it on the Cancer Genomics Cloud platform. So this is currently a public app, and if you search it, you're going to be getting the CTD app back, and it's going to give you a brief background of what you're doing. You're identifying these highly connected F sets inside of a graph. So it's going to note that it was designed specifically for metabolomics. But I would say you can also use this to look at transcriptomics data, definitely, as well as proteomics data. You just want to make sure that whatever your inputs are, they're well normalized across your studies. So for example, we use log2 normalization for transcriptomics data, but that's in the paper. And if you need help, you're welcome to contact our lab. So right now, what your inputs are is either an adjacency graph, or you can, if you don't want to build the graph yourself, we actually have a function in the CGC app that will build it for you. So all you need to input currently is your experimental and control data sets. So if you have, for example, transcriptomics for an experimental data set of Alzheimer's patients, say, and then normal healthy controls, you just need to input those tables. Just make sure that they're normalized across the two. And then here you can also input your nodes of interest. So say I have a list of 20 genes I'm particularly interested in. I can find exactly what's highly connected to those genes. Or you can do an approach that's similar to what we've done in this paper, and then just import those large WGCNA modules that you'd like broken down further. So here you're going to have outputs as your set of highly connected genes. We've actually set it to the p-value threshold is 0.05. You can play with this yourself if you'd like to in the app. I think there's an option to change it. And it's also going to export the graph. So if you import an adjacency matrix or your experimental control data sets, it's going to export an iGraph. So you can play with this graph further. And this is kind of going to help you visualize where these highly connected sets of genes are going to be. So this is what I'm excited about, but we're always in the process of improving these apps. We're always in the process of extending CTD. So the first thing we'd like to do is pre-compute knowledge graphs. So a question that we have is, if you have a set of genes, how highly connected are they in no knowledge graph? So for example, KEGG, string, or reactome. The problem here is that these graphs are going to be very large. So say 8,000, 16,000 genes and highly connected. So because CTD diffuses from each gene to every other gene, this is going to take several hours to compute. We're trying to do this ahead of time. So the ideal use case is the users will be able to just click select KEGG, input your genes of interest, and in 2 or 3 minutes, you're going to know exactly what's highly connected in KEGG, exactly what's highly connected in string, and how they compare to one another. We're also looking at developing out this guilt by association function. So because CTD is diffusing from each node to every other node, what you're going to get is you're going to get the nodes you're interested in. So say you start with the purple nodes and you want to ask which ones are highly connected. You're going to get the nodes that you're interested in, but you're also going to get these bridge nodes in between. So these are nodes or genes in our case that by including them in this module, you're going to decrease the p-value of the specific subset and that are going to be highly connected biologically to your genes you're already interested in. So we've actually tested this anecdotally on a use case with Alzheimer's disease. And what we're able to find is if you do a training test set and you leave out a test set of genes from your training set, you're able to rediscover these genes later on using this guilt by association function. So we're very excited about this, and we think it'll be particularly useful for example, discovering new disease genes or helping you refine your targets if you have a very large group of genes you're interested in. So with that, I'd like to take any questions specifically on the CTD app, how it works in the paper, or how you can apply it on the CGC. And I will mention that the CGC application is much easier than installing locally with Python or R. There's many less dependencies you need to deal with. You just need your inputs and that's it.
Yeah, so say I have just my log2-fold matrix out of RSEM or something. I can just bring that directly, or?
You can bring that directly if you have a disease and a control version.
Okay.
Because we're comparing the two.
It is comparison based.
It is comparison based. So I didn't go as much into detail on this, but the way we do this is you have your experimental and control, and you build one graph off of both of them. And you build another graph just off of control. And this is actually built into the CGC app, so you won't need to do it yourself. But you're going to prune the graph built on all samples based on the ones built on control. So you're essentially trying to eliminate signal that's there, but doesn't have anything to do with your experimental condition.
Cool.
So you can import those straight. You don't need to adjacency. If you have an adjacency matrix, we can build off that too. It just depends.
I'm also curious about this this guilt by association algorithm. The image here makes me think that you're kind of asking, like, are there genes that we have to traverse across this node multiple times in order to connect our module? Is that generally how that's working?
Yes, yeah, exactly. Are there genes
really
that you need to go through in order to make this a tight, highly connected module? So if you leave them out, you're going to lose your connections, right?
Yes, I see.
Yeah.
That's very cool.
Thank you.
I don't mean to take over all of the question time. Does anyone else have any questions they'd like to ask Varduhi?
And I would like to say, if you're trying to use CTD either as a local install or on the CGC, please feel free to reach out to our lab. We'll see what we can do to help you out with that as well.
Okay, well, I think I'm going to call it there if there are no other questions. Thank you so much, Dr. Petrosyan. Really appreciate it. Very looking forward to having this increased functionality on the Cancer Genomics Cloud for CTD.
Thank you. Yeah, thank you for having me today.